Our next speaker is Bill McClain, who's the Halverson Professor of Molecular Biology at the University of Wisconsin. So Bill has long used elegant genetic and biochemical approaches to study the maturation of specific RNA species, in particular tRNAs. And his pioneering work defined the complex multi-step pathway, how a transcribed DNA turns into mature tRNA. He's also made major contributions to our understanding about the specificity, about how the DNA sequence is converted into the amino acid sequence that determines protein function, which is obviously the fundamental process of translation. Sid and Bill were postdoctoral fellows together with Sidney Brenner and Francis Crick in Cambridge in the 1960s, and they began their work on tRNA processing at that time. And so Bill has followed this field very closely, and as part of that interest in history, he's written a fantastic summary of Sid's discovery of the enzymatic activity of RNA-sp. I recommend it to all of you. It's in the Journal of Molecular Biology, and I think we'll have hear a little bit of that today as part of his talk. Bill?
I want to go over here. Put the microphone over here. I feel particularly honored to be here today to celebrate this great occasion of the significant accomplishments of Sidney Altman. Congratulations, Sidney. It's been my great pleasure to know you for nearly 50 years, and I'm among many distinguished colleagues, and I appreciate that too. I'm very humbled by it. Today I'd like to talk about the origin of life on the planet Earth. And I hope to convince you the process is so simple, it's probably going to have happened elsewhere in the universe and beyond. Perhaps not generating human beings, but the mechanism by which the code arose is extremely simple. As we understand it, the RNA world generated molecules that became proteins, and proteins are the predominant, protein molecules are the predominant determinants of an organism's behavior and shape and function. And proteins arise from the genetic code and transfer RNA. And I was thinking about the two, and I think they're inextricably linked. Neither the genetic code nor the transfer RNA has any meaning, biochemical or genetic, without the other. Just think about that. That's really true, that neither construct has a meaning without the other. They go in unison, and they make protein. And that's what I'm going to talk about today. So I went to the web right away when I was invited to this, and look what I found. I found a postage stamp celebrating 100 years of the Chemistry Nobel Prize. And I think we know this fellow right here. He's sitting in the front row over here, Sidney Altman. So congratulations, Sidney, yet again. And also, I found this slide, Sidney giving his Nobel lecture in Stockholm. And I found this. There's his Nobel Prize, his medal, his plaque. And each of the laureates that year got a painting, a very beautiful original painting. And I'd like to point out one thing. Tom Cech was also awarded the Nobel Prize for catalytic RNA. However, only Sidney discovered an enzyme. Tom Cech discovered a catalyst. Well, Sidney discovered a special kind of enzyme, one that turns over multiple times, doesn't destroy itself. And so I think this is an enduring statement. I think all of you know this statement by Francis Crick. It's not escaped our attention that the specific pairing we have postulated suggests a possible copying mechanism for the genetic material. That's the or that's the discovery of the DNA double helix in 1953, published in Nature as a 2-page article. And then some years later, except for a trivial reason, novelty is a trivial reason, we should not be surprised that RNA is an enzyme. Well, that's really great. What a profound statement. So we have the genetic code, 64 triplets. We have transfer RNA. They make protein. This is the genetic code. You see the gold amino acids. I'm going to try to convince you today those were the first 5 coded amino acids by an early genetic code. And it's so simple the way they were found genetically. Proline is a central player, and I'm going to tell you why. Proline, not, you know from the Chow and Fasman, it turns corners. Its conformational space is very limited, so it's important to make a hairpin. Why do you want to make a hairpin? I'll tell you why. You want to make a container for mass action reasons, so reactants can find each other, proximity and orientation. This has never been discussed previously. So just a brief review is to attach an amino acid to transfer RNA, you first activate it with an enzyme, an amino acyl tRNA synthetase, which I abbreviate here as AARS. There are 22 amino acids now. When we were students, most of us anyway, there were 20. Since then, selenocysteine and pyrrolysine have been found. So there's 22 amino acids. Each has its own synthetase, each has its own tRNA, and they're activated so they can be transferred to the end of the tRNA in an active form, so they're prepared for protein synthesis. The amazing thing that Dino Morass found is there's 2 classes of amino acyl tRNA synthetases, and they approach the tRNA molecule either from the left side, and this is the Rossmann fold, parallel beta strands and flanking alpha helices, and this is the right side. This is the Morass fold, and it's anti-parallel beta strands and alpha helices in an approach. It's a dimer. These are all monomers, and this is a dimer. You know what dimerizes? You know what the key amino acid is? Proline. Proline is the key amino acid to dimerize. If you don't dimerize, you don't amino acylate the tRNA substrate. And there's 10 members of this class and 10 members of that class. It's really remarkable. It's 10 of each. This is at the beginning of life. This happened way back at the beginning. And it's, of course, been maintained. Look at the tRNAs. They attach these synthetases. I've kind of flipped them. This class 1 here attaches the amino acid to the 2' hydroxyl of the terminal adenosine of the ribose of the adenosine. And class 2 attach it to the 3' hydroxyl. Now going into the ribosome, they have to be on one particular site, and there's a way to convert that. EFTU catalyzes that reaction. But this is remarkable, really, that all systems, all genetic systems that are known proceed this way. So there's 10 members. I haven't put the other 2. The 2 that are missing belong in this class 2. And here are the 5 I'm going to tell you were the original 5. And Gilbert Ariani, and this is a student of Dino Morass. He was a graduate student. And Marc Delarue was the person who had the real insight. They found this proline separated by arginines with a certain number of residues between them. And that was sufficient to pull out from the PDB 10 proteins. And they were all these class 2 synthetases. And then, so here, here we have, there's probably more than 1,000 PDB files of class 1 with the Rossmann fold, dinucleotide reductase, parallel beta strands. But there's only 12. There's only 12 of these. This is the original. This is good. I'm going to try to convince you this is the original synthetase. The way it worked was by separating the strands to look inside an A-form helix. You can't see the edges of the bases on an A-form helix. So it ran out of space. You separate the strands, pretty soon you run out of space for specificity for adding new amino acids. So you got to figure out another way, and that's where class 1 came in. Now, in 1988, by computer analysis, I identified a subset of nucleotides for each of the 20 tRNAs. By I mean me and my group. Hugh Nicholas, a statistician, I had a half a dozen statisticians, a subset of nucleotides, and we found for alanine, tRNA, and E. coli that a GU pair was distinctive of it. Now, Paul Schimmel, working independently with Yaming Hao, made a similar observation that a GU Wobble pair near the 3' acceptor end was responsible for the acceptor activity. So this is the standard cloverleaf structure of a tRNA. And this is taken from our science publication. There's the GU pair. Now, originally, Francis Crick introduced the term Wobble. It was an information exchange group, IEG. Do any of you remember those? You got them free from NIH, just send it in. So it was information exchange group. And then it was published in the Journal of Molecular Biology. We see at the top here the standard 2 Watson-Crick pairs. You see the glycosidic bond, the same angle. You can flip them, and they're perpendicular to the axis, 90 degrees. Whereas a Watson-Crick pair, the G goes down into the minor groove, reducing this glycosidic bond angle. And the U is pushed up into the major groove, increasing that angle. So Paul Schimmel's interpretation is that the enzyme, the amino acyl synthetase, is just looking for this amino group in the minor groove. You get 3 kilocalories from that interaction. And the specificity for amino acylation, just the first step, is 10 to the minus 3. I mean, the error rate is 10 to the minus 3. From 3 kilocalories, it's not possible. So we had a different interpretation. And ours was that the GU caused a helical irregularity. That irregularity is what I'm going to talk about for the remainder of the talk. This was published again at 88. We knew right away, the minute we saw GU, we knew Francis's structure. We also know that Olga Kennard showed that AC was isosteric with GU, in that the A is pushed into the minor groove and the U into the major groove. She was studying DNA. She was interested in mutagenesis. So this paper was published several years later. Paul Schimmel criticized our work very seriously. He said, you can't trust genetics. You know, you can get these serendipitous reactions and serendipitous results that have nothing to do with biochemistry. So what we did is we translocated this GU pair to an adjacent site. So if GU is causing a helical irregularity, then if you transfer it, it should retain some of its function, right? On the other hand, if the protein is looking for that 2 amino group at a specific spot, it might not work. It's going to be moved 4 or 5 angstroms away. So here's the result. This was done with a nonsense suppressor, reporter protein, with the marker, the nonsense mutation of position 3. This is the read-off from the protein sequence, methionine, N-terminal methionine isoleucine. And look, 100% alanine there at the 3rd position by translocating the GU from position 3 to 4. So clearly, this supports our proposal at the time, and we've subsequently done a lot more. And I'll show you a crystal structure that will convince you still more. Now, we had this sequence for 30 years. And I noticed once when I was very ill, I had a hip replaced after waiting 30 years. Look at this GU here, but look what precedes it, GCU. You know what that is? That's an alanine codon. And it turns out the last U is part of the GU pair that determines alanine going on the end of the tRNA. Well, what a coincidence. What a coincidence. So I've noted that here. So GCU, actually GCN, where N is any of the 4 nucleotides, makes an alanine codon. And then on the other strand, so you read the codons 5' to 3'. You all know that, of course. at 158 bacterial and archaeal alanine sequences to see how frequently we found GU at this site here, and then subsequently we looked over here to see what was there. Here's what we found. At this position here, where the GU, at this position here, where the GU, you see what we found here? I have to come around here to read this. We find serine, a wonderful hydroxyl group. We find alanine. We find glycine, a great turner to turn a helix to make a hairpin. Proline, the interface of the dimer of the synthetases. Of course, this is the evolved synthetase. Do people in the audience know that the tripeptide N-terminal proline, threonine will catalyze an aldol condensation reaction with the same Kcat and Km as a 400 amino acid residue protein will? The two people that discovered that got a Nobel Prize in chemistry in about 2001. So proline is pretty important to have it, not only dimer interface, but it was catalytically active very early on. And we see another threonine, there's a hydroxyl group, you see it in an aspartic acid, and then it asymptotically approaches 0. And then if we look on the other strand, it's running in the opposite direction, so you would expect different amino acids. But here we find alanine and glycine as the major players here. Really quite surprising. As I said, we had this in front of us for 30 years and never paid any attention to it. So we wanted to ask, are these two distributions different statistically? Obviously, visually, impressionistically, they're different, but we subjected it to statistical analysis. So hypothesis A is the histograms represent the same distribution, and that's the null hypothesis. And then hypothesis B, they're different distributions. And we found that the null hypothesis is rejected, and the risk associated with rejecting that is 4/500%. So they're really now statistically different. Not only impressionistically different, but they're statistically different. So now John Sutherland, MRC LMB, current guy, he got the Gregor Mendel Award, by the way, from England for this work. What he's found is that all the reactants that were necessary to start life were present at the same time with cyanide. They didn't appear one at a time, but they were all present. This was published in Nature by Patel. John Sutherland is the last author. So the ribonucleotides, the amino acids, lipids. Lipids were very important. And so Mike Yaris here, he's shown just with a 29 nucleotide RNA, right here, you see that? This is an acid gel. So this is deacyl tRNA, and this species migrating more slowly is this strand right here with phenylalanine that's been attached to it by this 29 nucleotide RNA. It runs slower because of the increased mass from the phenylalanine, and then the positive charge on the amino group makes it run slower in the gel. So not only were the reactants there, but they were active. So here we have O-linked polysaccharides. Sutherland found the precursor to this. Well, how could they act? They could, right here, they could attach serine and threonine by their hydroxyl groups to this. Well, why is that important? They could make a cage. What's the cage good for? It's a precursor to a cell. For two molecules to react, they have to be in proximity and they have to have the right orientations. It has to be something before the cell. The cell is very complex. So what came before the cell? A cage, probably formed through threonine and serine attaching to the lipid. And they were in a hairpin loop with the proline kinking the peptide, as we understand it from current protein structure. That's a hypothesis, of course. And so I've just put these little hairpins down here. And you'll note that This is, I call it alanine proxy. We have tRNA alanine knockout cells, and so we're able to isolate variants of the tRNA. And the variants, again, now we see here, they have glycine, proline here, glycine, serine, and proline. They still code for the same amino acids. So here we have the, these are the 5 amino acids that were first coded. They all can be accommodated by just G and C. So maybe the early codon was just G and C and later incorporated A and U. So it started with 8 codons instead of the 64. 2 to the third power is 8. And you can get the other 2 introduced just in 2 filio generations by incorporating mispair. Wobble pairing, you can get a G pairing with a U and A pairing with C. So it's possible to get from this to the 64. Now, in these knockout cells that we made, we found the most active variant. The most active variant when you substitute GU is CC. Who would ever guess that? You can't do this in a knockout cell because the anticodon in a knockout cell, it's going to go to glutamine or lysine as an opal suppressor. But we found this, it's almost the same as wild type. So my colleague at the MRC, at the LMB, Gabriel Varani, is a spectroscopist. And so he determined the NMR structure of this tetraloop. First he did the wild type tetraloop, and then he did the tetraloop with the CC. He did it in collaboration with us. And so this is the NMR spectrum of the CC tetraloop. And you'll see the base pairs, the entire structure is identical for this CC tetraloop, except for the CC. It's in rapid equilibrium, millisecond to microsecond time scale. That means it's coming apart. You know, it hasn't got a defined structure. He lowered the pH to stabilize the base, to try to see possible base pair interactions. The helix fell apart. Well, just think about this, the beginning of protein coding. If the helix can come apart, that'd be a great way for a synthetase to inspect. So there you are, it's lowering it, it came apart. Now this is B-form DNA. This is the famous Watson-Crick. And you see how the bases, if we look here, this is the helical axis. The bases are at 90 degrees relative to that. In A-form, this is an A-form RNA helix. The edges of the bases aren't exposed, so it's not possible for a synthetase to look and discriminate between different potential acceptors, because you have to take the strands apart, at least partially, to have a look at them, to see what they are. But helix opening, thermodynamic stability is not sufficient in and of itself. Strand separation is not sufficient in and of itself to specify or to get amino acid alanine attached, because we have GU with a TM of 87, GC is 100 essentially. This is an active mutant. This is suppressor, nonsense suppressor efficiency, 80 degrees. And here you see C, 78. Here you have UC. It's inactive. It still has this low TM. So strand separation isn't sufficient in itself. It's much more complicated than that. So the local separation of the strands then is probably what's happening. Then I had a student, Hunsik Choi, who made a library using knockout cells where he randomized all these positions. All these positions are important for the tRNA alanine to accept alanine. Left this one alone. This is where RNase P cleaves. We didn't want to fool with that. And he got this remarkable mutant right here. It's shown in red. All these bases changed. This is mutant called 7-1. It has the same growth properties as the wild type tRNA, changing all those nucleotides. Well, they must act as a group. They must act as a group. The minute I saw it, I'm sure Hunsick realized this. He wouldn't talk. He grew up within a mile of the DMZ. He wouldn't say anything. He was taught you survive there by saying nothing. So I told him to change each one of these bases individually back to the wild-type base here. When he did that, it killed it. Just as if you were mutating the wild-type tRNA from an A to a C, you kill that. So these are acting as a cooperative unit. Well, that's all we knew. We didn't know more than that. But you'd never learn this by doing single nucleotide mutagenesis. You wouldn't learn it from doing crystallography either of a synthetase tRNA pair. You wouldn't know that these nucleotides were acting as a cooperative unit. So Dino Morass and I tried really hard to get a crystal structure. So this is the class. These are the 10 class 2 synthetases. You see, they're superimposed on the synthetase and the tRNA. See, here's all the 9 of the tRNA. Here's alanine over here. It's so different from the rest, the other class 2 synthetases. And now I'll show you in the last slide, the helical the irregularity that we're talking about. The yellow is the backbone of the free tRNA, uncharged, and you see it clashes with the synthetase, but in the form that's bound to the synthetase, right here, the amino acid form, the major groove is widened by as much as almost 3 angstroms. That's gigantic. A base is 3.4 angstroms. So it's a gigantic. It's all along the helix all the way. There's all 7 base pairs in the stem and into the T stem. So now, that's why I say the genetic code, tRNA, and protein synthesis are inextricably linked. And so the last question, I didn't know that Matt Messelson was going to talk about sex. But Sidney asked me the question, well, where did the codons come from? Francis Crick says you can't pose the right question until you're near the answer. Well, we think we know where the eggs came from. Did the egg come before the chicken? Which came first, the chicken or the egg? It's really a profound question. If the egg came first, where did it come from? If the chicken came first, how did that happen? Well, we're on to that. Now, I'll hope to talk to you again in the future. My collaborators, Hugh Nicholas, a postdoc for 10 years. He went to Pittsburgh Supercomputer Center after that, and now he's back as a postdoc. And Guy Plunkett got his degree with me. He's at the Genome Center. He collected all those sequences, 100. There's so many errors in the tRNA sequence in the database, the DNA and RNA. There's so many errors. You got to be an experienced person. And so without him, this project would not have happened. And incredible financial support from the National Institutes of Health, General Medical Science, incredible. And then, of course, my stipend at the present time. I'm an emeritus professor, and I'd like to thank you for your attention. Thank you. We have questions.
How do you get that support? What's the trick?
I'll tell you the story. No, I want to answer that question. You do the experiments before, you get the answer, and then you write the proposal as if you hadn't done the experiment. So then the study section, when they review your continuation application, they see you've done everything you said you're going to do, but you've done it ahead of time. Of course, you have to have a running start, so to speak.
You always tell me we already did it.
The study section says they already know it? Yeah. Don't propose an experiment that's too good that they're never going to forget it. If you haven't, unless you've already done it. I once made a proposal, really a clever experiment, but it just wasn't feasible. Because nucleic acid bases, they have a preference who their neighbor is in a helix. The rules for this aren't known, but I'll tell you, they have bases they like to be next to and bases they dislike to be next to. Those rules the physical chemists haven't figured that out yet. We tried to overcome it when we made these libraries. We couldn't do it. IDT, DNA RNA synthesizer. You can't get a random sequence. We couldn't. We tried hard.
Well, wouldn't you know that from the distribution of the
No, I mean, when you make a synthetic RNA, when you order it from a machine chemically synthesized, you put in an order. You don't get back what you order. I'm telling you, it's not a random distribution. Some bases don't like to be next to each other. That probably underlies part of the genetic code, if that's your question.
No, my question is, wouldn't you detect the fact that you're mentioning, the fact that certain guys don't like to be next to each other, and the distribution of amino acids and whole proteins?
So the question is, given my claim that some bases don't like to be next to others, wouldn't that impact the distribution of amino acids that we see in the genetic? Yeah, of course it would. But what would you like to predict what that is? What that would be? You first have to know what the rules are. The rules aren't known. There's not many people that are aware of this. No, I'm talking about messenger. I'm talking about genome sequences. I'm talking, all DNA, all RNA. This is not limited just to tRNA.
Additional questions?